Hello, my name is Jackson Huff, and today I will be describing to you the process that is known as CRISPR gene editing. Many of you have heard of CRISPR gene editing through the news and other sources. I believe that it is very important that we know about CRISPR gene editing, seeing as though it can affect us and the lives that we live. For example, many of us know a farmer or may even be farmers ourselves. GMOs, or genetically modified organisms, take advantage of CRISPR and allow the genetically modified organism to have modified genes that may make it grow better, or some other farming implement that allows for genetically modified organisms to be grown a little bit easier than the non-genetically modified variant of that organism. Today, I will be explaining to you the history of CRISPR gene editing, the definition of CRISPR gene editing, how it works. Then I will show you a visual representation of CRISPR gene editing in action. And then I will give you how the process works again in my own words in a fun little way that should help you remember exactly how CRISPR gene editing works. CRISPR gene editing was discovered in 1987 in a biosafety laboratory in Japan. Researchers in the laboratory were trying to discover exactly what made bacteria so resilient. As you and I both know, bacteria are very resilient little creatures. We all have to take various precautions to make sure that we are not getting infected by these bacteria, and we all also own cleaning supplies in our own homes that kill these bacteria. Researchers were focused on E. coli bacteria, and according to the Journal of Bacteriology written by Dr. Ishino, CRISPR is actually part of what makes these bacteria so resilient, seeing as though CRISPR makes up a vital part of the bacteria's immune system. When a virus attacks a bacteria, the bacteria actually rewrites the DNA that is contained within the virus. The bacteria makes it so that the virus cannot do any harm to the bacteria itself. Scientists could not figure out the exact process in which the bacteria rewrote the virus's DNA until approximately 2007. By 2007, scientists had a very firm understanding of CRISPR and how it worked inside the DNA of viruses and how bacteria used it, so much so that they were able to move on to human trials and produce human genomes with modified genes that were modified by using CRISPR. According to Dr. Wayne Hunter in his work called CRISPR-Assisted Targeted Delivery into Adult Ovaries, CRISPR is actually an acronym that, when broken down, is very easy to understand. Going letter by letter, it starts with C. The C in CRISPR means clustered, and when you think of clustered, it means something that is tightly packed together. Like the genes in the human genome, there are tight bunches of genes that all control multiple gene representations within people, and they are responsible for certain traits. Next, R, and R means regularly. As you and I both know, if anything is regular, it can happen often, or it can also mean that it happens normally. The reason that we can study CRISPR is because the genes happen the same way over and over again. Next, I, or interspaced. If something is interspaced, it means that it is spread out evenly and equally. Interspaced simply means that the genes are spread out. Next, S is for short. Short means exactly what you and I believe short means anyway. It means the gene itself is expressed in small or a short amount of space, much like we would say that somebody that's short is expressed in a smaller amount of space. Now P stands for palindromic. Now you might think that palindromic is a big scary word, but all it means is that it is the same forwards and backwards. Think about the word mom. These genes are just like the word for mom. M-O-M is the same forwards and backwards. These genes are also the same forwards and backwards. And the last R in CRISPR simply means repeating. CRISPR works by identifying genes that show up over and over again, and these genes that we would say repeat. Take, for example, the gene for blue eyes. Everyone with blue eyes has the same gene, meaning that it repeats. How does CRISPR work? Dr. Kadek and Iskra laid out exactly how it works in their work titled Target Heritable Mutation and Gene Conversion via CRISPR. RNA first cuts a section of DNA. Then the RNA gets rid of the genes that were in that space, or you can think of it as the RNA throws away the old DNA. Then the RNA puts itself exactly where the DNA used to be. The RNA acts like a simple tool that we use every day. Think about cut and paste. RNA simply just highlights the word like we would, and then it puts a new word or a new piece of gene information into that spot. The RNA puts the new piece of gene information much like we would put a new word into the sentence. And then, much like a sentence gains a new word, the cell gains new genes. Now I will have a visual representation of CRISPR. Watch how it begins with a large open space, then the genes are removed. Now that they are removed, new genes enter and take the spot that the old genes were in. After the new genes take their place, the DNA molecule reassembles itself into the familiar DNA molecule that we are all used to. And that is basically how CRISPR works. In summary, recall that CRISPR was discovered in a Japanese laboratory that was trying to understand how E. coli fight off infections so well. This was done in 1987. Next, think of CRISPR as cutting and pasting small bits of information into the cell. Remember what I said, where we cut and paste a word into a sentence. Think about that word that we are putting into the sentence being like a gene that we are putting into the cell. We take out the word that used to be there and we put in a new word. Just like CRISPR, where we take out the gene that used to be there and we put in a new gene. Finally, remember that the goal of CRISPR is to change the genes within the cell, and that's what it's all about. The whole point of CRISPR is that we can take the genes that were in the cell before, edit them to the way we want them, and then change exactly how they are. Are there any questions today? Thank you very much and have a great rest of your day.